it automatically fuses and creates this process, but it's actually a complex cellular fusion aspect that it uses a thing called syncytin. And that is actually present in a lot of our cells, if not all of our cells, not all of them, but a lot of them. And so that's why COVID can hijack ACE2. And I think when we talk about risk, we talk about risk with these viruses, or even bacteria, because some bacteria can do this too. When we want to talk about the capabilities of a virus, it comes down to which of our cellular abilities it can hijack. Can it hijack our ability to release cytokines? But can it fuse cells into other cells? Is something not all viruses have the capability to do, or bacteria, or pathogens. When we talk about this, when we talk about this risk, we're discussing if they have the ability to hijack our cells' capability to form syncytia, then that is the most dangerous, and the speed at which it does it then to determine dangerousness from there, and then the outcomes, does it make like Ebola does it, but like it does it differently, much more rapid. So it's just trying to understand this from an objective standpoint, I guess, like we were talking about earlier. We can objectively discern different types of cell destruction and the timetables and pathways, and you can relate those into objective scales that we can relate to one another. And then we can create relative risk integers based on determining these variables and breaking down. It's like that's how you objectively prove risk. That's why saying that COVID is objectively closer in risk to HIV than it is influenza is simply because of its ability to form syncytia that influenza does not have. So, and this is where with H5N1, we see some blood vessel problems that imply it might be forming syncytia. So there's some concern in my mind that H5N1 has similar capabilities, but I'm not willing to go out on that limb yet. We'll see, but it's something we should definitely keep an eye on.